in 2,000 children are born with epilepsy, and this can be the result of misspellings, basically, or errors in a single gene. And in most of these cases, we don't really know why or how these faulty genes cause epilepsy. And it's highly likely that these faulty genes are likely to be occurring early on before birth. But obviously it's very difficult to study these early developmental stages. To try and investigate this further, I have developed a system whereby I've been able to take small portions of developing brain tissue and study this in the laboratory. These samples are donated from a very special biobank here in Newcastle called the Human Developmental Biology Resource. I will use these samples to be able to assess how the faulty genes are affecting early brain development in human fetal tissue. So I'm going to use this new approach that I've been developing here in Newcastle, and I will alter genes that are associated with epilepsy and see what effect this has on the developing brain. And this is really exciting because I think we'll be able to look at the underlying cause of this disorder, but also a major advantage of this model is that it's highly adaptable. So we can use this to investigate multiple different genes that are associated with epilepsy. The most immediate impact this will have is that for families and people with variants, for example, in STXBP1, we'll be able to give clarification and understanding for the mechanisms that underlying the disease, essentially. And I think that's crucial for then being able to go on to understand the next treatment options that we will go on to study. But to be honest, I think that's only going to be the start, because the next 5 to 10 years beyond that, I think, is where we're really going to see the impact of this model system, because I'm now starting to prove its value with this particular gene of interest, but as I mentioned before, it's got a highly adaptable nature that we can actually look at multiple genes of interest. So I want to go on to look at many genes and then also use it as a platform to see whether we can actually reverse any of the effects that we observe, or even just to use it as a screening tool for multiple drugs that are being developed at the moment to treat epilepsy.